Okay, thank you very much. That was really fascinating. So we're going to go on now to talk to Megan, who is going to give us a presentation on immunology aspects.
Thanks very much, Sheila. I'm going to share a couple of slides with you, but I'll start by thanking the organisers for inviting me and also everyone in the audience for joining us this evening. My field is immunology, as Sheila said, and I care about very much about the T cells that Tess introduced to us. And I kind of wanted with these slides to give you a bit of an overview of our understanding of vaccines and how the amazing knowledge that we've generated in the last almost 100 years really has enabled us to design better vaccines that are more protective. Many of you will be aware that the first vaccine that we had in the world, the first formal vaccine, was against the smallpox virus. And this was using a related virus, cowpox, to protect people. And this vaccine had absolutely no help from our knowledge of immunology and no help from our immunology of virology, because we knew really nothing about those two fields back in the 18th century. And all of these data were based on observations that infection with cowpox protected you against the more deadly smallpox. And really, going forward a large amount of time into the 20th century, we didn't make a lot of progress in how we made vaccines. Up until the 20th century, we were still using variants of the virus, so modified versions like the cowpox virus, or killed pathogens to work as our vaccines to protect us. What did happen in the middle and later 20th century was that we started to understand more about the viruses, and we started to understand much more about cells and molecules in our immune system that protect us against disease. And just one big advance that I wanted to highlight to you was our ability to identify correlates of immune protection. Tess has just told you that she spent a long time developing antibody methods in her lab to be able to see if people are making immune responses. And that's exactly what we're looking at here. So here, I'm just giving you the example in the context of flu. And this is the hemagglutinin protein in flu, which the virus uses to attach onto our cells. And if you have a vaccine that can drive an antibody response that can bind to hemagglutinin, you can now prevent the virus from attaching to our cells and from protecting against infection. So now we can know what the molecules are that vaccines need to generate to protect us. And that means that we can test whether a vaccine is likely to be protective. So Tess underlined to you the importance of the trials they did in animals before they moved on to humans. And it was this type of antibody response they were looking at to show that the vaccines were likely to be protective. I wanted to highlight to you here 1986, which is a really important year in vaccinology. This was when the hepatitis B vaccine was launched. And this was new and exciting because it wasn't one of these old style killed or modified viruses. This was a single protein taken from hepatitis B, and it was combined with some molecules that induce our immune response. So our ability to identify what protein was needed from the virus needed a lot of knowledge from virologists. Our ability to know how to stimulate an immune response required a lot of work from immunologists. And then we also needed help from general cell biologists and protein chemists to enable us to make that protein to then put it into a vaccine to protect people against that disease. So now we have the skills and the knowledge to actually design vaccines in a much more planned way so that we could protect people. As Sheila's already pointed out to you, the platforms that are being used in the COVID-19 vaccines, for example, here in the UK, rely on these platforms that appear to have come from nowhere, but actually people have been working on the underlying science for many, many years. So viral vectors generally have been in use since the 1980s and were first used in the plant field to generate recombinant proteins. They're used by us scientists in the lab for our discovery science. And now, as you know, they're also used for vaccines. And that can be vaccines for us, vaccines for livestock, and vaccines also for companion animals. The mRNA technology, as Sheila said, people have been working in the context for many years. And back in the 90s was when they were first tested in the context of driving a protective immune response. It has taken us a long time and a lot of work from a lot of researchers to be able to tweak the messenger RNA to make sure that it's stable when it's injected into us and to make sure it induces the right type of immune response to give us that immune protection. So I hope you can see that we started off with vaccines that relied not at all on our knowledge about immunology and virology, and then moved on with an acceleration in our knowledge over the 20th century to be able to really design the best types of vaccines to protect us. I was asked to talk to you about viral variants and the concerns that they are being raised. I wanted to start by making sure that we all were aware of what the concerns are in that area. This is an image from a science paper from last year, and this is the viral spike protein that Teresa mentioned earlier. And this is a part of the virus that binds to our host cells via this protein ACE2. And the first type of mutations that we're worried about are those that happen right at that part of the virus that binds to our host proteins. And that can mean that the virus gets more sticky and more likely to infect us. What happens when we're infected or when we get a vaccine is that we make this neutralizing antibody. And what that does is it binds to the same part of the virus, this receptor binding domain of the spike protein, to stop it from binding our cells. So now the virus can no longer stick to our cells and no longer cause an infection. And the second type of mutations we're worried about are those that happen at this area where the antibody binds to the protein. And then that might mean that the antibody is less able to bind to the viral protein. And that what we're seeing from the variants that are starting to circulate around the globe is that the neutralizing antibodies in people who were infected with the original virus or people who are being vaccinated are slightly less good at binding to and preventing infection. So that is a cause for concern, but I think we have to remember that neutralization isn't all that antibodies do. So they're very good at triggering immune responses and helping to control the viral infection. So they may not be as good with the new variants at stopping infection, but they're going to allow you to control the infection, so you're less likely to get disease, and also you're less likely to make lots of copies of the virus and transmit those to other people. The other important thing to remember, Tess also mentioned these guys. These are the T cells, that other arm of your adaptive immune response. And generally we think of them as CD4 and CD8 T cells, and these can kill virally infected cells, and they coordinate this very tailored immune response that means that you can control the virus infection. And in the past, it's been really difficult for us to measure the T cell response in humans that are infected or vaccinated. But we've developed a lot of tools and techniques, often starting in animal models, that now allow us to track those T cell responses to the virus. And that's led to a lot of really powerful work in the last year, showing that both virus infection and vaccination leads to these CD4 and CD8 T cells that are specific for the SARS-CoV-2 virus and that we think are protecting us. And I think the thing that's really important about the T cell response is that each individual, when you're infected or vaccinated, will make several T cell responses. So you'll have several T cells responding, and that means the virus is less likely to escape if it has mutations. And while most of us will make a very similar neutralizing antibody response, we'll make quite distinct T cell responses. So that will make it even harder for the virus to mutate away from our distinct T cell responses and be able to infect lots of people in the community. We know that the vaccines are inducing these cells, and it looks like that they are able to recognize the variants more or less just as well as recognize the original SARS-CoV-2 virus. So we should be worried about viral variants, but we do need to remember we have these other layers of protective immunity that will hopefully reduce disease and reduce the chances that the virus replicates inside us and transmits to other people. I'm just going to end with a couple of thoughts. I'm a fundamental or basic scientist, so I care about how cells and molecules work. And these are two of the big areas of questions where we still don't understand the fundamentals of immunology that would really help us to design even better vaccines against SARS-CoV-2 and many other types of pathogens. And those are in the B cell field, and how we can make vaccines that induce very long-lived cells that produce these protective antibodies. Some vaccines induce cells that only last a couple of years, some for the lifetime of the entire person. If we knew what signals drove those long-lived ones, then we could make vaccines that were much more durable. And if we could understand how we can make antibodies that are less susceptible to viral escape, then we can make vaccines that are much more likely to give us protection in the long term. In my lab, we care a lot about T cells, and a big question is how we can produce long-lived T cells and have them present at the sites of infection. Those of you who've had your vaccine already will know that you get your vaccine in your arm, whereas you get infected with SARS-CoV-2 in your respiratory system. And it's going to take several hours to days for those T cells to move into the infection site to start protecting you. So if we can understand how we can make better vaccines that are delivered to our mucosal system, then we may be able to produce even better vaccination to protect us even more quickly, to reduce disease and reduce transmission. So I'll stop sharing there. Thank you very much.
Okay, thanks, Megan. I wondered if you might like to share what you think about any possible requirement for booster vaccinations in the future.
And so I guess boosters come in two ways. Many of us will have had tetanus boosters that are needed because those antibody forming cells don't last very long. So they are boosting the same immune response. And we're going to have to watch over time people's immune responses to see how well the vaccines are at keeping those plasma cells or antibody forming cells alive to give us that durable antibody. The other concern, as I've said, are the variants of the virus. Many of you will have had a different flu vaccine every year, and that's for exactly the same reason, that the flu mutates and you need to change your antibody response or update it to the new variant. And I suspect we're very likely to see new vaccines for SARS-CoV-2 towards the end of this year or next year that will have those variants that are more prominent in the world and have those spike proteins present inside those vaccines.
Thanks, Megan. That was really clear. Great.
Thanks, Sheila.